Today, together with my team, we are presenting a study published in BMC Genomics that investigates the genome sequencing of canyon-type yaks. Yaks, or Boscronians, are cornerstone livestock of the Qinghai Tibet Plateau. They sustain pastoral communities by providing meat, milk, fiber, and transport. What makes them remarkable is their ability to survive in extreme conditions such as hypoxia, cold temperatures, and limited forage availability. Canyon-type yaks that inhabit deep valley also show distinct morphology and production traits compared to the plateau yaks. Therefore, studying them provides valuable insights into genetic adaptation in harsh environments. Most genomic studies have focused on comparisons between wild and domestic yaks, but breed-level differences remain underexplored. In particular, canyon-type yaks have not been extensively studied despite their unique ecological conditions. This creates a clear research gap. Understanding their genetic diversity and adaptive trait is crucial not only for conservation and improving productivity, but also to identifying specific SNP markers that are useful for breed identification and also molecular breeding programs. This study was guided by four main objectives. First, to resequence 225 canyon-type yaks in order to generate a comprehensive SNP dataset. Second, to evaluate genetic diversity and population structure to clarify evolutionary relationships. Third, to detect genome-wide selective sweeps, identifying regions under strong natural or artificial selection. And finally, to identify candidate genes linked to key adaptive traits such as hypoxia tolerance, growth, and reproduction. Together, these objectives show a logical flow from generating data, analyzing diversity, detecting adaptation, and finally connecting genes to the traits. This completes the introduction, and now I will pass to my teammates to explain the methods.
Assalamualaikum and hi, my name is Nurul Fadila binti Muhammad Azali, metric number A203029. Next, I will proceed with the methodology of the research. First, sample collection and sequencing. A total of 225 domestic yak from 3 populations were sampled. Blood, ear tissue, and hair root sample were collected for DNA extraction using the standard phenochlorophore method. The extracted DNA was then sequenced using Illumina Novasec 6000 and HiSec 4000 platform, with generating 115 base pair rates with approximately 10 times coverage. Sequencing quality was then assessed using FastQC and MultiQC to ensure high-quality genome data for further analysis. Next is genome mapping and SNP calling. About 2,597 gigabytes of sequencing data were processed and low-quality reads were removed using Thermomatic. The clean reads were aligned to the yak reference genome using BWA, followed by duplicate removal and quality recalibration. SNP were then identified using GATK pipeline and filtered to obtain the high-quality SNP dataset for further analysis. After SNP identification, additional filtering was performed to remove low-quality variants and missing data. Population structure analysis was conducted using PCA, phylogenetic trees, and AgMisha to study clustering pattern, genetic relationship, and ancestry composition among the yak population. This analysis helped to evaluate genetic diversity and population differentiation. Next is selection sweep analysis. This study used nucleotide diversity, FST, and XPEHH to identify genomic region under natural selection. Regions with extreme value were selected as candidate region under selection and visualized using Manhattan plot. These steps helped to identify genes involved in environmental adaptation. And lastly, the identified SNP were mapped to genes and analyzed using gene ontology and KAGG pathway analysis to determine their biological function. Statistical correction using the Benjamin-Hochberg method was applied to improve result reliability. This analysis helped explain the biological roles of selected gene in yak adaptation.
For the first results of wide genome resequencing, we were able to see the Q30 score is more than 93% high, and the GC content is around 43% to 44%, while the duplicate rates is less than 10%. And this shows the alignment between the yak reference genome, and this further approved by the SNP intersections. And we see that it's around 20 million high-quality SNPs of intersection between the 3 yaks. Okay, on the population structure results, PCA method shows that gene Chuan is the most concentrated. You can see the red circle on the graph showing the gene Chuan yaks, and this indicates the lower genetic diversity of the gene Chuan yaks between the 3 yaks. Furthermore, on the population structure, we look at the lowest CV error, K equals to 3, And we see the pattern there is that it's very different pattern between the three yards. And this indicates the three distinct essential differences of the three yards. In addition to that, the unit SMP shows the Ank yards, Gauyan yards, Jinchuan yards have different concentration, especially the Jinchuan yards has distributed across the genome. And this indicates the broader genome diversity of the Jinchuan yards. And this may result from the structure variance of the yards, historical selection events, subject to population-specific selective pressure, and demographic processes. On the third results, selective sweeps, we use three methods, which is pi ratio, FST, XPE, H4. And total of the loci that we able to select is around 6,411 loci target selection region. And this distributed primarily on chromosome 6, 16, 20, and chromosome Y. After figuring out the loci, we do the gene annotation, in which there are 13 candidate genes that we are able to analyze, in which it shows a few characteristics like growth and development, metabolic regulation, neuronal function, productivity and reproduction, transcription regulation, and phenotypic regulation. And this reflects the yard's multi-adaptive evolutions. Furthermore, we look at the gene frequency by looking at the MAF value, which is the green, yellow, and red. The higher the value, more common minor alleles there are. And this shows the region undergoes sustained and intense positive selections. Lastly, the genome function analysis. From GO function enrichment, on 13 candidate genes, we able to show 13 candidate genes. Its enrichment's multiple function terms are key role in neuronal signal transmission and synaptic structure. And they are also enriched in function related to GTPase activity, and this potential role in cell signaling, transduction, and energy metabolisms.
As for the comment, my first comment is about the SMPs identified in the study, which just like a barcode, this is significant because it allows us to develop SNP chips. Just like scanning a product at a store, farmers can scan the yak's DNA to verify its breed, purity, and see its survival potential instantly. Secondly, this research gives us great details about the yak capabilities. It shows that the gene like SPATA5 for the muscle and ITFG1 for male are the biological reason why this yak can climb steep canyons and feed their babies even when it's freezing and there's not much air. For the third one is, this research makes the breeding of yaks much easier because we can now find the best traits in their DNA. If a farmer wants a yak that grows fast but also produces a lot of milk, they can use these 13 genes as a guide to pick the best parents for the next generation. In conclusion, this research is very important because from the DNA of 225 yaks now, we get to know how they have evolved to survive in difficult canyon environment. By finding 13 special genes and creating a barcode systems, scientists have given us a clear map to help both animals and the local people. This work makes it so much easier to protect the unique yak's breed and help the farmers produce better food, ensuring this animal to stay healthy, strong, and keep producing good offspring for a long time.